Hello everybody, this is Dr. Sharkar here. Very good morning my dear students. Today in this video I will be giving you some informations and we'll be discussing about a very interesting topic is plasmid, right? Before starting with this plasmid, I will ask you, or rather I will remind you that the DNA is a very important thing which is already known to you because I have already discussed about DNA elements and the chromosomal DNA also. But the plasmid, this term is little bit of unknown to you, right? Therefore, I will say that the plasmid is not very much unknown to you as because the plasmid is also the DNA element, but it is in a different form, right? So I will start with plasmid by describing its definition, right? The plasmid is a genetic element in cell which are able to replicate independently. It means they are not dependent on the chromosomal DNA for its replication process because the plasmid do contain the origin of replication, which is very important for replication process. Right? Okay. The plasmid is a very small DNA which are covalently closed circular form and they often found in bacterial cells. Other than bacterial cells, they are also existing in the eukaryotic cells also, like yeast cell. Okay, fine. Now I will move into the next section is the different forms of plasmids. It means the classification of plasmid. How do we classify plasmids? First of all, there are different forms of plasmids which can be classified as F plasmid. Secondly, the R plasmid. Third one is cholecynogenic or col plasmid. The fourth one is the TI plasmid. Fifth one is the linear form of plasmid. Sixth one is 2-micron plasmid, which is found in the yeast cells, right? Okay, so now move on to the next portion. I mean, we will describe about each and every individual forms of plasmids, right? Okay, so first, what was the first one? The first classified form of plasmid is the F plasmid, right? What is F plasmid? The F plasmid is a kind of plasmid which is comparatively large plasmid, right? And around the size is around 100 kilobase. And it does contain two origin of replications. Okay, fine. These kind of plasmids do contain a gene which is known as tragene. It means this particular gene allows the plasmid to transfer between cells. It means the plasmid bearing cell will be able to transfer its DNA from one cell to another cell, right? Now I will be giving you an example of these plasmid bearing cells. E. coli is a very common bacterial cell which is known to everybody, I guess, right? So this E. coli is a cell which often contains this kind of F plasmid. And this F plasmid is commonly known as fertility factor. Okay, fine. So why is it? Because those bacterial cells do contain these kind of plasmids. They are regarded as F plasmid because F fertility factor bearing cells. And now if once this particular cell having the criteria or having the this particular F plasmid, they are now able to transfer its plasmid DNA element to another cell. The cell, it means the cell doesn't contain the F plasmid, right? Now, once the F plasmid is being transferred from F plus cell into F minus cell, now the F minus cell will be transformed into F plus cell, right? I hope and I guess till this end you understood this matter very well, and I will provide you the slides, PowerPoint form of slides to you till this much, and it will be very much understandable by every individual student. So don't worry, the PowerPoint presentation will be given to you and you will be able to study it properly, right? Till now, this much, and in the next video I will be talking about the different other classes of plasmids. Okay, dear students, thank you so much. Okay, have a very nice day.